So on all the written slides with the blanks, go ahead and just pause the video and fill it in, and I'll probably skip most of that. So this is really important. You understand the condensation, hydrolysis reactions. If you look here, you have two amino acids, two separate molecules. They're coming together. Condensation, you're building. And you see this peptide bond being created. Peptide. It's between the carbon and the nitrogen right there. And you'll see this water left over. And that's where it gets the name condensation. You got water left behind, kind of like put a can of soda out. It's cold. It's going to get water collecting on the outside from the condensation. And then when you break it apart, it's called hydrolysis, water splitting. You need to add water to it and split them back over. But if you keep doing this, keep building more amino acids, H, carbon, you're just going to keep more and more and more. You're going to build a polypeptide. All right, so you got to be able to recognize those molecules. So yeah, here you go, carbon, essential carbon. We got an R group, nitrogen, hydrogen. Here, instead of the H, it's going to go to another nitrogen. And you kick out an H over here. Or actually, no, on this side you kick out an OH, and on this side you kick out an H. And when you make this peptide bond, you create water. Now let's finish drawing this other amino acid. We got an R group. Okay, so make sure you can draw this. Okay, draw two amino acids coming together just like you saw in that earlier slide. Pause it, keep drawing it. You gotta know how to do that. So basically, 20 different types of amino acids. I didn't know until not that long ago there's actually more amino acids than that, just not that are used in living things to make proteins. And you got a bunch of different kinds of amino acids, and really what kind of amino acids you have will change the shape of that protein because they have different chemical properties. So these ones up here in the yellow, those are all nonpolar. And down here, these are polar. So these will not mix with water still. These will fold inward with their inward. Let me rewrite that. So if they're on that long amino acid chain, where we have all these long amino acids stuck together, if they're there, they're going to try to fold in and get away from the outside. And your cytoplasm is water-based. These polar ones, they're going to face out. So if you have a mutation where you go from a polar to a non-polar amino acid, you could have a major change in that protein, change the shape, and then it's not going to work like it would otherwise. Then you have also not just non-polar, but you have different charges. These are all positively charged up here, and the other ones are negatively charged. So positive, these are negative. And again, if you go from a positive to a negatively charged through a mutation, you're going to have a potentially different amino acid or differently behaving amino acid polypeptide chain and a different protein. But it's pretty cool to see. These are all the different R groups. Some have these rings. Here's double rings, single rings. Some don't have any rings. You can see the R group is highlighted with the yellow and then the blue here. You see the main part of the membrane or the amino acid is the same, but very cool. I like seeing this. All this different. Simplest one is glycine, just an H, just straight up, just an H. Serine's pretty simple. Carbon, OH. You don't need to memorize that, but I like showing that. Okay, so any kind of sequence, and you have gradual, all this complexity because you have all these different choices, 20 choices for each position. You have 500, amino acid long polypeptide, you're going to have a whole lot of different potential ways you can build that. Collagen, I like collagen, making your skin strong. You see it in lotion bottles on the ads a lot. Improve your collagen and put this lotion on here. And so, yeah, you see these wrinkles here. That's from just the collagen not working as well over time. And that's what makes those wrinkles. I think pretty much that's how it works. That collagen is not grabbing a hold as well anymore. Here's a very important protein. This is hemoglobin. Hemoglobin in your blood or on your blood, part of your blood cells help grab onto oxygen. And as you know, those of you that have given blood, they test your iron levels. And that's a test of your hemoglobin, basically. They want to make sure you got plenty of hemoglobin on board before they take your blood, because you're going to be compromised in terms of oxygen carrying capability. One time, or not one time, but maybe like 6-7 years ago, the Red Cross kept calling me. Every 2 months I went in for probably a three-time period, and that's not. It takes 90 days to really rebuild your blood supply, and they're getting me every 56 days and 2 months basically. And they wore me down. I actually tested low on my hemoglobin when I got my every once in a while blood test. And they're like going, "You're losing blood. Where are you losing blood?" I'm like, "Oh no, I'm not bleeding." You're not losing it. If you're losing it internally through your intestines, your feces will be black from the broken down blood, and that wasn't happening. Turns out the Red Cross was stealing my blood. So here's an interesting, a cool example of a protein that has two polypeptide chains. This is inactive integrin. It's kind of floppy there. And then it gets activated. So this is active. It'll create a signaling. See how it changes the shape? Boom, it changes shape. It'll grab onto this and cause a whole cascade. It's called a cascade of reactions in your cell, which then create whatever function or action it needs to perform there. Okay, so yeah, we got globular and fibrous. So either shaped like this or shaped like this in little globs. It is what it says pretty much. And just some good, diagrams to show you it's collagen, fibrous, globular, or myoglobin. So yeah, fibrous. Pretty easy to remember this. Globular, fibrous. Examples: collagen, I keep talking about. Elastin, keratin as well. And then hemoglobin. And then your antibodies is another big category of globular protein. So go through this. Make sure you have paraphrased. You'll see in the book this list of different types of proteins and their function. Pretty cool. Tubulin. I love that word, tubulin. Your microtubules that attach to your chromosomes. The tubulins are on there that help the chromosomes can pull themselves apart from each other. And then cool blood clotting. Proteins can turn into gel when they're activated. It's pretty cool, changing chemical structure and their physical state basically. Proteins here, this would be hemoglobin and cell adhesion. They seem to stick to each other. It has some different hormones that are proteins, although most of them that we think about hormones are steroids, but they can be either histones, receptors, immunity, I mentioned, antibodies. And you have this, we know about the genome, right? The genome is, I think I had it up here, all the genes. A gene is all the genes. Oops, I messed that up. Genome, good thing I checked here. The genome is all the genes of the cell, tissue, organism. And your proteasome is another thing that's very important and that we're learning more about all the time. And you can see these with electrophoresis. A couple different cool. Rubisco was one of my favorites. We see this when we learn about the Calvin cycle. Help to take carbon from the atmosphere as a gas. You make a chemical product with it, sugar, pretty sweet. Insulin, you know, that's in diabetics. People that have diabetes need to have that insulin given to them, type 1 anyway. Immunoglobulin, antibodies, rhodopsin, oh, so sweet. In the retina, changes shape, creates a nerve signal once light hits it. It's pretty awesome. Our collagen we talk about, spider silk, very strong. We need to be able to know about denaturation from pH and heat mostly. Go ahead and pause it to fill everything out. And a good way to think about it is an egg. You know, an egg white is clear, liquidy, and then as you cook it, as it denatures, it changes characteristics quite dramatically. And that's irreversible change. Denaturation should go here. Okay. And yeah, this is the egg, what it looks like. It's pretty cool. And this, you may have heard of Botox. What people get. People are getting that stuff young. 30-year-old people are getting Botox now. Can't stand one little wrinkle, now you have Botox everywhere. Anyway, what it does, it interferes with nerve signal transmission. It prevents acetylcholine from being released. So normally it's released acetyl, abbreviated ACH here, and then you have a signal. Well, you get that Botox, the signal can't go and the muscle can't contract. So people, like they talk, they're going to go like this. They can't really smile. Basically causes paralysis. Very mild paralysis. If you get a big shot of this stuff, you're dead. You're going to be dead because your heart's going to stop. Your diaphragm is a muscle that keeps you breathing. It's going to stop moving and you're going to be dead.